Welcome to the Church of Perpetual Life. Hey, glad you're here. My name is Neil Vandry. It's my pleasure to be your officiator here tonight, and I'd also like to welcome everyone who's joining us in the live stream that we are doing tonight. In past services, we've had over 60 people joining the live stream, and they're coming from all over the world, all over the country. Many people are having what we call watch parties, arranging watch parties in their homes or in their offices or in restaurants. They're watching the live stream, they're having potlucks, or as I say, going to restaurants and doing what we're doing here. So there may be hundreds of people, we don't know who's watching, but we're glad to have them. And if there's anyone out there that would like to do a live stream in their hometown, contact me and we'll help you to coordinate that. Now, I want you to be sure to be here at the end tonight because we have a really big announcement to make tonight, a surprise on the advancement in medicine that Bill is going to discuss at the end of the meeting. You're going to want to hear this. It's really amazing. Here at the Church of Perpetual Life, we refer to ourselves as immortalists, not because we have defeated death yet, but because we have faith in future technologies to conquer disease and aging, as well as death itself. While we fully acknowledge that this technology does not appear to be currently available, the impressive history of human problem solving and technological advancements give us faith in this inevitability. But it is with sadness that I have to let you know that our assistant, our previous assistant, Stacy Marie Swift, has passed away. And so we want to acknowledge that. We're very sorry to see that. She had an interest in cryonics but did not act on it. So we're very sad in the passing of our beloved Stacy Swift. You may have remembered her being at the front desk. You may have received phone calls from her. So sadly, she has left us. And we're going to have a memorial on her also next month. Now, you may have heard about RADFest. RADFest was a couple of months ago, and many of our members attended to see presentations by people such as Ray Kurzweil, Jim Mellon, Dr. Neil Reardon, Terry Grossman, Suzanne Summers, Aubrey de Grey, and our own Bill Faloon gave presentations to people. There were about 1,000 people from all over the world who came. These were immortalists from all over the world, and now they have the recordings available from RADFest. You can get your copies of these videos. And Doug, I think we have a short video on this. If we can show the video on the RADFest.
Welcome to RADFest 2018. Great to be here with people from literally all over the world. People who are pioneers in your own right, who are not accepting the limitations of the mortality paradigm and are willing to do something about it. We are excited to have this time together. What makes this event really, really special is the exceptional quality of people that are involved in this event. Yes, and it's the people you're going to see up here, the thought leaders and scientists and innovators, and it's also the people that are in the room who are experts in your own right and pioneers in your own right who are bringing the quality to this event. So thank you so much for being here. And finally, RADFest is a party. Because we're here to celebrate. We're here to celebrate life. Hey, if you can't celebrate life, you can't really extend it, can you? So we're here to celebrate life. RADFest is divided into 3 segments: Near Horizon, Next Horizon, and Now. Near Horizon is going to cover all the areas of innovation that are happening that are in the sort of human trial phase. So modalities that are coming to you in the near future that it's very important for you to know about. Next Horizon is really about a vision for the future. What does our future look like? If we're going to stick around, it's important for us to have a vision of our ageless, deathless future, and it's important for us to understand that it looks very different than our reality today. So that's what the Next Horizon is about. And finally, the third section is Now. What we can do right now to build our longevity, which will be consummated with a distribution by Bill Faloon of Life Extension Foundation, the first ever age reversal guide. which is a huge milestone in our revolution because we have age reversal to be guided to. So we're excited about that. And by the way, I want to let you know that RADFest is presented to you by the nonprofit Coalition for Radical Life Extension. And yes, the coalition. And the coalition's mission is to put on events like this as part of a larger effort to galvanize public opinion in favor of radical life extension and to see resources go to making a difference in challenging the limitations of aging the way we want to see it happen. So that's the good work that the coalition is doing. Now I want to introduce to you your hosts for this event. James Stroll is co-founder and co-director of People Unlimited. He's also the director and creator of RADFest. And Bernadine is co-founder and co-director of People Unlimited, his partner. Please welcome James and Bernadine.
We're on a mission together. We're on a mission to send this powerful information, everything that we're doing here together, around the world. That's what RADFest is about. It's about the power of togetherness, the power of sharing the information that is saving lives. And that's what this is about tonight with each other. Being together, sharing the information, inspiring each other to take good care of ourselves. Like Joe said, Faloon will be sharing information about modalities, therapies that you can take home with you, information now that's very powerful. Many others will be sharing that information here, this entire event. But to even get there, to get where we want to get, you have to take good care of yourself now, strengthen yourself. That's what Rad Challenge was about here. I want to encourage all of you to begin to take on that mindset that brings the future to us. Look, our future is bright. No one here, as far as I'm concerned, has to die anymore. No one here has to end up in some frail state where they can't move their bodies. We can all do something about this. Let's encourage each other. Let's cheer each other on to life. Cheer each other on to life. You know? Really, you know, we're all important in this. We're going to hear top scientists, wellness doctors, enthusiasts here, thought leaders, like Joe said. But all of you are important in this. We all play a part because this is a movement. I want you to get this. This is a movement. We want to move this movement across the world, and every one of you are important in this. I'm so grateful that you showed up for your life here. Spread the word to everybody on this planet, everybody you meet, that we're doing something very special. Guys, we're creating a whole new world together, a world without aging, sickness, and death. So I'm thrilled because information is powerful. Information, but most of all, being able then to share that information. I say all the time, I've said it every RADFest, and I'm going to say it now, I think it bears repeating continuously, that we shouldn't be fighting each other on this planet. Because our worst enemy is not each other. Our worst enemy is sickness, aging, and death. We should be working together on every level, sharing that information, getting into great discussions. There's no better discussion than how can we stay alive, how can we get healthy, to inspire each other in that. If you're not with people that you're sharing that information with, I want you to get with people. RADFest is great. You know, we're all here. We're doing this. We're going to do this for 3 or 4 days. But what do you have after RADFest? What do you have when you walk out this door? I want to encourage you to find people that you can really share this inspiration with. And tell them then about the next RADFest coming up. We're building momentum. We are building momentum. You can feel it. You can feel it here in this room. We've sold out the exhibition hall here. We've sold out this hotel. We've got a huge amount of people that are here in this event. Next year will even be bigger. We are responsible together, each and every one of us, to making this new life happen. I think that's exciting. I think that when we really get in our bodies that we're in charge, that we have something to say about our well-being, that we have something to say about our lives, like each and every one of you do here. How long do you want to live? How healthy do you want to live? Because the exciting thing, and I was telling some news people today, it's not that, you know, look, we're not talking about living longer in some decrepit state. We're talking about living longer with vitality and strength and energy. I feel better today than I've ever felt. I feel more excited about life. I had one of the news people ask me today, well, wouldn't that be boring, living forever? I said, I don't have a boring moment. I said, sometimes I pray for a boring moment. It gets more exciting every day, life does. So like I said, I'm so excited about my partner, Bernie. We were just talking. You wouldn't believe she's now going into her 82nd year on the planet. We don't... But she's ageless. Bernie and I, we don't use the word old. We say we've been here for so many years. Yeah. And we need to be inspiring each other. Yeah. Do great things. Stay alive. Stay alive for each other.
Yeah. Hi, everybody. I'm really, really happy to be here with all of you. It's really exciting to have this many of us come together with real purpose in mind, and that's outliving death. Yeah. I'm radical about everything that has to do with staying alive. I'm radical against aging and death. I don't think we deserve it. I think we should live abundantly forever. Those are my feelings. And the greatest success there is not dying. That's the greatest success. I'm inspired. I think we should be inspired. I never lose inspiration at all. I'm excited for every day that comes. I'm looking forward to it. I'm looking forward to what's going to happen 10 years from now, 100 years from now. I'm looking forward to it. I plan on being around. I didn't come to die. I came to live. And so join me. Let's do this together. We need to be together. You know, look, people are going to think it's weird that you feel like you can live a long, long time, if not forever. They think it's weird. Well, you know what? I don't care anymore. I'm out to really benefit in what is happening. Be proud of who you are.
I want to tell you that everybody in this room has an opportunity. I don't care what condition you're in, the body is miraculous and it can be turned around.
Yep.
But I want to encourage you to start doing that now. I want to tell you though tonight, it's very important to take on the mindset of being here forever or for however long you want to be, for super longevity. It's very important to take on that mindset now, to see yourself there, to begin to bring the future to you. Because all these therapies and all these modalities are going to work a lot better if you embrace them. If you physically and mentally embrace them. Make yourself ready physically and make yourself ready mentally. Begin to get out of the old age consciousness. Begin to think, get out of the old age program, the death program. Begin to do things that are way out of the box and being alive. So I want to encourage all of you to spread the word about don't be afraid to tell people anymore. Look, it's happening. There's all kinds of information on the news now about radical life extension, all kinds of great therapies coming out more and more, and information about the ideas of super longevity. We need to be proud here in this room of feeling this way, and we need to tell everybody that we meet that there's another way to live. There's another way to live. Thank you all. Thank you all very much.
Go ahead and put that last, is it possible to put that last clip on? Doug, if you can. You know, there's a movement sweeping the world, the unlimited lifespan movement, and by being here, you can be part of this movement. You saw what was going on in San Diego at the RadFest, 1,000 people meeting, but there are thousands and thousands of others who weren't there around the world working toward age reversal, and I'm very happy to be a part of that. And I would encourage you to let your friends, the people that you know, that you love, to come here to learn more about age reversal. We have cards downstairs that can help facilitate that, little business cards. You can give them an invitation to come here, and those other brochures that we have that will tell them about what we are doing. I encourage you to tell all the people that you care about, let them know about Perpetual Life, and highly encourage you to get the videos from this RadFest, from this just a couple of months ago. And go out there next year. It's a wonderful thing where you're going to get inspired, you're going to get information, and you're going to make connections with people in age reversal. Our next service will be here at the church on December the 13th, Thursday, and we'll have a presentation on cryonics by the president of the Cryonics Institute coming down from Michigan, Dennis Kowalski, and we'll get an update on age reversal from Bill, December the 13th, Thursday. And of course, we'll also have our annual Remembrance of the Resurrectable ceremony to honor the people, all of the people who are currently in cryonic suspension. As usual, our doors will open at 6 o'clock and our presentation will begin at 7. One more reminder about our library at the outside this sanctuary, in the what I call the outer sanctuary, we have a library on age reversal books. You're welcome to take one, check it out, and bring it back next month. Our speaker this evening comes from upstate New York near Syracuse. And we have a short film to show you a little about his company and the work that they're doing toward radical life extension. I had the opportunity to drive around South Florida with this man who spends a lot of time in the snow, and we got a chance to see the beach. And just earlier today, there was a boy, he's about this big, and he's carrying an iguana that's about this long. I said, "Is that your pet?" He said, "No, I just caught it." So only things that you see in South Florida. But let's go ahead and show the Ichor film, Doug, to show a little bit about Ichor.
From my perspective, Ichor does something that's unique for most of the companies I've ever been involved with. They take other people's ideas that have failed and they find the reason why they failed, and then they make them succeed, which is really sort of neat because that means somebody had a really good idea but didn't know how to implement it. We implement it, and that's what Ichor does really, really well.
I either had to do medical school or do research. I couldn't do both in parallel. So I independently applied for and won a research grant that allowed me to set up a small company in my living room in an apartment building. And that's really where the company started.
Ichor is such a large thing. It's more of an idea than it is just a soundbite. We have many portfolio companies. So we're really a company of companies, and we use our contract research organization and our income from that to seed new risky ideas that normal people wouldn't want to go after. And from doing that in a systematic way, we're really driving a lot of value in the space of biogerontology.
Nice little clip there, Kelsey. Kelsey Moody is a biotechnology pharmaceutical executive who specialized in the study of aging for over a decade. Since 2013, Kelsey has successfully built Ichor Therapeutics from a living room startup into a premier research organization that has raised over $20 million to support drug discovery for pathways of aging. Kelsey has received graduate level training in medicine, business, and laboratory research. Please help me welcome to the stage, Kelsey Moody.
Testing. Alrighty, thank you, Neil. Welcome everyone to the Church of Perpetual Life, and thank you, Neil, for such a kind introduction. It feels a little awkward and odd for me to be in a church. I'm a biotechnology and pharmaceutical executive, and I'm used to presenting at academic conferences and that sort of thing, so the venue is a little bit unique for me. But what I think this really points to is a need that's recognized increasingly so by the mainstream pharmaceutical industry, as well as everyone in this audience, and that is a need for more effective, targeted therapies to go after the diseases of aging, not just individual disease states, but also as general holistic processes. It's also a great thing to run into Neil again. I first met Neil almost a decade ago, or perhaps slightly more than that, at the Undoing Aging conference in UCLA, so there's always a little bit of nostalgia whenever Neil's in the room, and he's been a great supporter of this entire movement over time. I chose the title, A Coming of Age, Longevity Becomes Mainstream, for this talk, because I really think what I see in the field and what I think a lot of other people are seeing is a real renaissance of new ideas for targeting age-related pathology. Just 5 years ago, when I was still a medical student at Upstate Medical University, which is excellent from the perspective of being a traditional conservative medical institution, the idea of trying to go after aging as a conserved process really wasn't discussed. It was kind of this weird thing that no one really talked about. And just 5 years later, not only is Ichor Therapeutics taking off 10 minutes south of the university, but they've just announced they're breaking ground on a longevity institute, which I think is very reflective of the attention that's now being driven into the space. Before I dive into the actual presentation, financial disclosures, everything that I do is for profit, so recognize that as I discuss some of the research we're doing, and I'm also an inventor on several active patents and patent applications that are relevant to my work and the work of others. I should also discuss a little bit about the team. Everyone thinks that they have the best team or says that they have the best team. I really do, and what's great about my team is the translational nature of that group. So a lot of people talk to me, they're like, why does it take so long? We all know this from the media, right? Why does it take so long and cost so much money to move something that works in cells or works in mice into a product that can be delivered for people? Why does it cost billions of dollars to do that? And I really think that that boils down to difficulties in communication between the different parties. If you look at the term itself, translational research, the process of going from discovery stage to something that anyone in the audience could take for a particular disease, translation means changing languages. The accountants and the financial and the business people speak completely different languages than the lawyers. Even the lawyers, you have transactional attorneys and you have intellectual property lawyers, and those are completely different languages than, say, the bench scientists that are working on the actual development of the products. Your prescribing physician speaks yet another different language, and so I think a lot of the underlying challenges in moving something from discovery to first-in-man studies is really based on a communication issue. And the teams that are very effective at this have great communication, and they have folks that can readily parse apart the different types of languages. So at our company, we have all of that, which I'm very thankful for. Aaron Wolf is our COO, who's a biophysicist. Our kind of running joke is if it's smaller than a cell, it's his territory, and if it's bigger than a cell, it's mine. Case Vortel's our CMO, and he's had a long and illustrious career in the pharmaceutical industry and is helping us move some of our promising programs towards the clinic. And then even on the legal side, David Reed and Dong-Hoo Son are our two in-house counsel that have really helped galvanize the team, and the list goes on. So we're very fortunate, despite being a small company, to have a lot of dedicated researchers as well as people that represent each of these different language hubs, if you will, to assist us with moving our programs forward. And then finally, of course, financial support. Ichor Therapeutics actually started because Bill Faloon and Life Extension Foundation decided to take a gamble on the dreams of a young medical student, and with a $500,000 placement into Ichor for us to get started, we've raised over $20 million, and we expect to do quite a bit better than that in the next year or two. We've also received financial support from a variety of sophisticated investors, a lot of angel investor support, as well as foundations that are probably familiar to a lot of you here. And for all that support, we're incredibly thankful. In terms of the talk itself today, there's kind of two different things that I'd like to discuss. The first, I'd like to frame how we as a company are approaching aging and approaching longevity. And then I want to take, in the second part of the talk, a little bit of a deeper dive into how we're approaching drug discovery in the context of longevity and anti-aging research. And I should say, I actually hate the term anti-aging research. We're not really anti-aging so much as we're pro-longevity. And this distinction is made in many talks, but I'd like to make it again here. We're not interested in necessarily making people just live longer in a decrepit and prolonged state. We're looking for fundamental disease processes that can be disrupted to increase youthful, healthy lifespan, and that's really what our company is focused on. So, the framework that we use to really set the stage for how we're going to approach drug discovery and aging is based on the SENS damage repair approach presented by Aubrey de Grey. And if you're not familiar with his work, I'll just recap it briefly here. This is Aubrey, and in his book, Ending Aging, which was published, I think, in September 2007, what he really did is outlined a roadmap for how we could go about targeting age-related disease, and it created kind of an action plan for for-profit companies to create products that were profitable and had viability in the marketplace, but could also advance actual anti-aging work at the same time. And the premise of his approach really boiled down to a pretty simple model. The idea is that in order to age, you have to exist, right? Existing is the thing that makes everything age. If it doesn't exist, it doesn't age. If it does exist, it almost always does. And for living things, existing and living means that you have metabolism. So the whole premise of his argument was that the things that allow us to stay alive, taking in nutrients, taking in oxygen, creating energy, and staying alive, are the things that cause us to age, get old, decrepit, and die. But the distinction that he made is that metabolism doesn't outright cause pathology. Metabolism leads to the accumulation of damage, different types of damage. And it is only after that damage reaches a critical threshold that pathology emerges and a disease state exists. Now, the conventional dogma for how to approach age-related disease really boils down to two different camps, the gerontology camp and the geriatrics camp. The idea behind gerontology is to pursue interventions that will reduce the speed at which metabolism drives the accumulation of damage. The problem with that sort of approach is that metabolism is inherently complicated. It's only partially understood. This is just a schematic of some of the major metabolic pathways that are known. But also interfering in this cycle, or in any of these pathways, has a real opportunity for off-target effects. So it's very difficult to get highly targeted drugs that are effective. That's not to say that none exist. I'm sure Bill has or will talk about NAD+ supplementation and infusions, for example, and a number of the different supplements and nutritional components that you're probably aware of are kind of a gerontology, health management, and damage reduction sort of approach. But from a drug discovery perspective, this is a very murky thing to try to operate in. Likewise, the geriatrics approach is really more, in my mind, about symptom mitigation than it is about disrupting an underlying disease process or damage process. And I liken it to having a leaky roof. If you have a small leak in your roof and then your ceiling starts to leak, you can address that problem by simply putting a bucket or a pan underneath the leak. But over time, that damage continues to expand, the number of leaks continues to expand, and all of a sudden it quickly becomes unmanageable. And from a drug discovery and development and from a physician's perspective, it's difficult to treat patients in this way. I suspect a lot of people in the audience are on several medications that they're chronically on for primary conditions, and then you're probably on even more medications to deal with the side effects of the first set of medications, and the list goes on. It becomes unmanageable very, very quickly. What Aubrey proposed is a third approach, and he calls it the maintenance approach. And the idea here is that you could periodically eliminate or remove some of the damage that accumulates, and that would allow you to periodically clean up the damage and prevent the damage from moving past the threshold necessary to create pathology. And by doing so, if you could identify the types of damage and if you were able to effectively remove them, you could perpetuate a state of relative youthfulness, assuming that your treatment modality was sufficiently comprehensive. What's very significant about this from a drug discovery perspective is that, and this sounds kind of bad to say, but the drugs don't actually have to work very well to be super effective. And let me explain why that is. When you're dealing with chronic conditions, this is a sort of a condition where the drug has to be administered on a very recurring, regular basis to patients. That means it needs to be very effective to support all the different types of ethnicities and genders and age ranges and everything else in the patient population. But what it also means is that the, excuse me, what it also means is it also needs to be a lot safer because people are constantly taking it. In contrast, if you have a very aggressive type of cancer, for example, the drugs that you can take for that can be a little bit more damaging because the concerns over the cancer are a lot more pressing, and that's something that you're probably going to take in a much shorter treatment window, which is why we get away with things like chemotherapy and radiation therapy in oncology. When we think about a damage repair strategy for aging, we think about these damages as something that takes an entire lifetime to accumulate. By extension then, a drug that works okay, that's used very infrequently, could have a lot of flexibility in a toxicity profile and also a lot of flexibility in an efficacy profile and still do a lot of good for the patient. So from a pharmaceutical perspective, this is kind of an interesting modality in that it presents kind of a non-chronic, periodic sort of approach that would be a lot easier to manage in a discovery and development process. And the way that Aubrey describes this, I actually stole this slide verbatim from one of his old presentations, but the idea of vintage cars. The distinction here is important though. Vintage cars were maintained to last, they were not built to last. And back to the damage repair strategy, you don't necessarily need to understand the redox reactions that lead to rust accumulation on a fender to scrub off the rust and repaint it, or when necessary, replace the fender. So there's a huge opportunity here to sidestep the gap in knowledge that we have about metabolism and still have a lot of effect in treating age-related disease. So this is kind of the motif that Aubrey presented, and in his book and through his foundation, he's presented 7 classes or types of damage that he believes are comprehensive in driving age-related pathology. Those include cell loss or tissue atrophy, so this would be the loss of stem cell niches and things like that over time. Cancerous cells in yellow, of course, we're all familiar with cancer. Mitochondrial mutations, so mitochondria are the powerhouse of the cells that creates energy for us, and like any other powerhouse or power plant, they do get old over time and need to be replaced or repaired, so that's one of the classes he's identified. Perhaps the hottest area of research right now is this idea of death-resistant cells or senescent cells, which we'll dive into a little bit more detail for later. Other damages include extracellular matrix stiffening, which drives tissue dysfunction, the accumulation of extracellular aggregates, so junk outside of the cell, and the accumulation of intracellular aggregates, or junk inside of the cell. Now, if we were to do something about a disease state as convoluted and multifaceted as aging clearly is, then a one-trick pony is not going to be sufficient to make a difference. So this was really the inspiration behind Ichor Therapeutics. What we've focused on with Ichor is to create a large company with wet lab research and full preclinical discovery through first-in-man capabilities. And with that entity, we've spun out a variety of portfolio companies, some of them single asset plays, some of them platform technologies, all of which focus on mitigating one or more specific damage categories associated with aging. And that's the way that we've been building out the company, and that's what our goal is. So of the various Ichor portfolio companies, we have Ichor Therapeutics proper. Fox Bio is our new joint venture with Juvenescence. Octus Biologics is an antibody mimetic scaffold company. Recombipure does protein expression work. And Toxerine does small molecule drug discovery. Lysoclear is a macular degeneration program. And Grapeseed Bio is our new strategic investment fund that's designed to fund other startup companies in the space that are looking to solve these problems for us. And we incubate those companies and work with them. The first group we funded is a startup called Repair Biotech. For the rest of the talk today, though, what I'd like to do is dive in a little bit more detail to our portfolio company, Entoxerine. And the reason for that is Entoxerine is as traditional a pharmaceutical, small molecule drug discovery sort of entity as that could exist. So I want to explain how we're applying some new protein expression technology to actually go after age-related disease and aging pathways. So we kind of have a rule at Ichor, and that is if you're doing something new, we don't like to do multiple new things at the same time. So if you're going after a new disease pathway, we want to go after that pathway in as traditional way as possible. If we're introducing a new platform or a new delivery motif or something like that, then we're going to go after pathways that are already established. And so what Entoxerine is doing is running a very traditional small molecule drug discovery program for proven longevity targets. And we're certainly not the only ones that are doing this. So the last year, 2 years, has been a huge explosion of interest in the idea of destroying senescent cells. Hundreds of millions of dollars and several IPOs have occurred around this. Probably the most famous is Unity Biotech. They're kind of the first major one in the space. Though Restore Bio is also working in the space, they work on immunosenescence. And for those of you that aren't familiar with senescent cells, they're frequently called zombie cells in the media. And the way that they work is as we age, certain cells in the body should be cleared out and destroyed, but they're not. They're allowed to linger, they're allowed to hang around. And over time, they accumulate in tissues, and pictures of them are here. They stain blue with a beta-galactosidase assay, so they make very nice microscope pictures for audiences. And as they accumulate in tissues, they secrete all kinds of pro-inflammatory factors, cytokines, things like that, that have a variety of negative paracrine and systemic effects on the body. They promote cancer metastasis, for example, and general organ and tissue dysfunction. But it's known that if you can selectively kill these cells, you can have profound health effects that are positive as a result of that. I don't have slides on it here, but one of the more recent studies with a combination of dasatinib, a leukemia agent, and quercetin led to a 37% increase in lifespan in mice that were treated at, I think it was either 20 or 24 months old, the equivalent of like a human 70-year-old. That's a very old mouse to be treated. And what's important about that whole treatment regimen is that wasn't something that they were on chronically. That was given a couple of times, and then it was stopped, and that was the effect size that was saw. It's quite remarkable. Perhaps familiar to some of you is this study that came out in March of 2017. It's a landmark study by Kirkland's, or De Keyser's group, rather, in the Netherlands, and they showed that blocking one specific protein-protein interaction led to profound regeneration in animals. This is the same animal before and after treatment. Again, this wasn't a chronic treatment. This was a treatment that was only given, I think, twice for this particular study. And what's significant is while we can appreciate that, yes, there's hair regrowth, and for all the guys in the audience, and shortly me, that's a fantastic thing, the animals weren't just regenerated from the context of growing hair. All of the major organ systems that were tested had improved function, functional output. So this is something that really led to a profound regenerative effect across the organism. Now, what I'd like to draw your attention to is this is a single protein-protein interaction. The way that this works is P53, guardian of the genome back in the 90s, if you recall hearing that going around. P53 is a protein. It should kill bad cells, like cancer cells and like senescent cells. Apparently, a largely unknown protein called FOXO4 binds it, And when it's bound, senescent cells are able to survive instead of die like they should. And de Keyser's group showed that using a peptide approach, they can disrupt that interaction, and this is the functional effect of that. And what's not appreciated by a lot of people is that, I can't say all, and I don't even know if I can say most, but many disease processes come down to very specific interactions of two proteins just touching each other. Sometimes they do when they're not supposed to, or sometimes they don't when they should. But a lot of disease processes boil down to that, and that's the basis for small molecule drug screening, either inhibiting those interactions or enhancing them. So let's propose a situation where we wanted to develop a new drug. And this is kind of a generic model. So say we wanted to make an anti-cancer drug or an anti-senescent drug, and we knew that two proteins binding to each other allowed the bad cell to survive. And just quick examples of this, Unity Biotech has BCL2 activators and disruption of p53-MDM2 interaction, and de Keyser spun out a company, Claria, that's FOXO4-p53, so this is what these people are doing. This isn't something I'm making up. So suppose we want to do that. Well, how would we screen for drugs that would prevent these bad cells from staying alive and clear them out? Well, what we would do is we would disrupt them with some sort of small molecule and hope that it kills them. And the actual roadmap for how we go through that process is as follows. First thing we need to do, I apologize, I changed my graphic here to the blue one and the orange one. The first thing we need to do is make very, very large quantities of the proteins of interest, and then we need to label them or develop some sort of assay so that we can tell whether they're bound together or whether they've been knocked apart. And then we need to screen hundreds of thousands or millions of compounds to identify compounds that can disrupt those interactions, and that's where drugs come from, and then you go through a whole iterative process after that. The problem with drug screening this way, and this is how all the pharmaceutical companies do it, the problem with drug screening this way is the fact that you need a lot of protein. And the way that we get a lot of proteins is to make them in microorganisms like E. coli or other types of bacteria. Those guys give us the scalability that we can't get if we try to make the proteins in human cells, but there's shortcomings. Bacteria lack the machinery to make many types of human proteins, because human proteins, many of them are more complicated, they're folded weird, and we'll talk about that in a minute. So microorganisms aren't able to make a lot of the target proteins that we would like to. As a result, the state of the art for many protein targets is to do fragment-based screening, where you make little pieces of the two proteins and study that interaction instead. Now, I think even though I don't think, are there any biophysicists in this audience? No? Okay. So even though there's no biophysicists in this audience, I think everyone can appreciate that if you're doing drug discovery on this, and this is how your target looks in the body, the results that you get here might not necessarily be translatable or applicable to results that you would get here. And the goal would be to make the full-size, properly folded proteins in a scalable system like E. coli or microorganisms, so we can do drug screening on the real thing and not something that mirrors it. Now I should say that for many different targets, the fragment-based screening, it has its place and it does very well for certain types of targets, but when you're dealing with new targets especially, where not a lot is known about the protein, you really want to try to do screening with a full-size protein if you possibly can. So we're going to do a little exercise in protein folding. So for those of you, just making sure we're on the same page, our bodies are made out of tissues and organs. Those tissues and organs are made out of cells. Within the cells, we're made of a bunch of stuff, including proteins, and proteins are kind of the business end of a lot of what we do. Proteins exist and are made in a linear form, where each little circle is an amino acid, and then they can fold either into a properly folded bioactive form, which is what they need to be to function, or they can aggregate into an aggregated mess, which is improperly folded, and that's a problem. So I need a couple volunteers from the audience to help me out with this demonstration, and I see everyone in the front 2 rows is instantly regretting their decision. Could I get 4, please? I've got both of you two, so we're going to take both of you two, if you don't mind. Come on up. And I need 2 more if possible, please. You guys look adventurous over here. What do you think? Can I steal the 2 of you? I promise not to embarrass you too badly. All right. Welcome to the stage, you guys. So, this is 40 yards of packaging tape, and this corresponds to our linear protein that we're going to make. And our goal is to make the whole protein, and then we want to fold it perfectly in half so that the stickiness is all gone. So we're going sticky to sticky when we fold this thing, okay? So I'm going to go along. Why don't we stand, you right here, please. You directly, yep, right there, and then equally spaced going down the line. How about that? You covering up your name tag so no one knows who you are. Lillian, fantastic. All right, so we're going to go ahead and make this protein. So why don't you just hold the tape right there for me? Great. And we're going to try not to let this fold on itself, okay? Because, you know, that's bad. So could you just hold that and make sure we're not making a mess of everything? Okay, this is great. And we'll have you grab right here. Excellent. We're going to go a little bit further. Howdy. And you'll hold there. All right, now just hold it like it is. Now, I think we can appreciate this is a packaging tape. It's really sticky stuff. Just like a protein, it's very easy to get on itself and kind of, so we're gonna say maybe right here is about the center. So we're gonna very carefully try to flop this over. So I'm gonna go ahead and come down with this. Let's very carefully try to fold it together into one piece. So hold on a second, I'm gonna start from here, and we're gonna come up this way. Go ahead and help guide it here. All right, and we can keep, you guys can, let's be friendly, let's all move in together. We're folding our protein, all right. I don't charge for Biophysics 101. All right. And voila, very good. You can let that go. And thank you guys, you can hop off the stage now. Thank you so much. We have made a properly folded protein, fantastic. So this is the protein as it exists in the body when made in human cells. The problem with E. coli is that they're lacking the wonderful support from our chaperone proteins to facilitate protein folding. Now, I am very good at this, so I got this folding thing covered. Let's say it was about that long. I exaggerate for effect, but only a little bit. So now we've got this protein, and shoot. I actually discovered this when we were trying to package a sample for one of our clients. And I was like, this is the best example of protein aggregation I can think of. And of course, you try to take it apart, and then it just, it's a mess, whatever. So this is what happens normally when you try to express a protein in E. coli or other types of microorganisms. You can run into issues where the protein aggregates, and you can appreciate that these two are not the same thing, despite the fact that they started with a similar piece of tape, a similar primary sequence, yeah? Now, the fragment-based approach, same kind of idea, you know, I can have a little piece like this, and I can replicate some of that functionality, and I get something that mimics some of the folding maybe, but again, this isn't the full size, so we're not mapping the entirety of the interaction. I'm enjoying, this is the first time giving this talk. I hope you guys like it. Now, I think we all appreciate that Mr. Bill Faloon is all about innovative, disruptive technology, so I think he should be our innovative, disruptive technologist. What do you guys think? Yeah. So this lanyard is actually my lanyard from RadFest. I was there, had a great time. So Bill, your RP tag, which is the technology that we've developed through Intoxerine to help us make larger fragments or full-size proteins for drug screening purposes. So I want you to stand right over there for me, and you're just going to hold this for me. And because we have For the technical people that might be in the remote audience, it's an enhanced solubility tag that allows the protein to fold. Though my packaging tape is not cooperative. Okay, that should be pretty good. And then what we're going to do, because Bill is there anchoring the protein, I can go with my lovely 7-foot wingspan and get us a nice full-size protein. Oh, don't let it go yet. We haven't added our TEV protease to cut that thing off. Should have asked Neil for scissors. There we go. And you're cut off, and now we have our protein. And that's basically how our technology works, and we raised $10 million for that. Thank you, Bill. Hi, Bill. So I hope that this little demonstration, though, allows you to appreciate the difference between full-size proteins that are properly folded, fragments, and aggregated nonsense that doesn't really give you reliable biophysical data. And for many different targets, and we're continuing to expand the scope of what targets we can go after, state-of-the-art is this kind of fragment-based screen, and where we are as a company with the drugs that, or the screens that we're doing, is really at these full-size proteins. And you can appreciate, again, by this demonstration and the graphic here, that those two really aren't similar. So why do we care, and how does this work in a real-world setting? Well, I should throw in, I promise this will be the only data slide that I actually include in here. So we can measure how tightly proteins bind to one another with a variety of different assays. This is a fluorescence polarization assay. And basically the take home is the further to the top left that the line is, the tighter the two proteins are binding. We have two proteins, A and B. The black line is fragments of both, and they bind each other with a KD of about 6 micromole, so that's very weak, bordering on non-specific binding. When we switch one but not both of those fragments to a full-size protein, we have an incredible tightening of the interaction, and the KD drops to about 270 nanomole. And then when we go full-size, KD drops further still to 114 nanomole, and we get the tightest binding yet. There is a difference of binding tightness 55 times. The full-size proteins bind 55 times tighter than the fragments do. So if we're doing drug screening and identifying small molecules that disrupt the fragments, that may or may not have any relevance to the full-size proteins, because the full-size proteins bind much tighter and in much different ways than the fragments are capturing, at least with these proteins. So if there's anything that you take away from my talk today, I want you to appreciate that better biophysics leads to better drugs. And this is something that's really been a paramount cornerstone for how we've built out this program, and we've had a great deal of success with it. I am going to show you a little bit of data, and I'm going to apologize in advance. The disadvantage of being a pharmaceutical executive is all of the cool stuff that I really wish I could share in this venue, I am not allowed to share, and I will get in trouble for. But I do have some teaser slides here that I think are pretty interesting. So what we did is, after we screened using our biophysical assay, we screened hundreds of thousands of compounds to find hits, things that passed our specifications in the biophysical assay, so that we could move them forward to try them in cells for the first time. I've got a summary slide at the end that shows all these images together, but the lighting's a little bit off, so I just blew up the images, and I'll show them one at a time so that you can see a little bit more clearly. So when we take normal cells and add the compound that we found in our hit, we see nothing interesting. We see typical cell morphology. The cells kind of elongate. They kind of spread out their cytoplasm on the bottom of the plate, and they're hanging out doing what cells do. And for anyone that's looked under a microscope, this is typical for what cells look like in a dish. And after 3 hours, no real difference. The cells are still doing their cell thing. We have maybe one little green spot here that could correspond to one cell dying, but maybe that's an artifact. In general, nothing to write home about. These two images are pretty darn similar. And I should specify that these are taken in the exact same, they're the exact same location on the plate. So the cells do migrate just a little bit, but this is comparing apples to apples, same exact images, just 3 hours apart. When we look at our senescent cells, compound added at 0 hours, same thing. I think just in the audience we can appreciate that this picture looks a lot like that picture, looks a lot like that picture. But look what happens when we add our compound that we found to senescent cells and leave them for 3 hours. Holy cow, that is a nuclear holocaust of dying cells right there. All of the green are different cells. They bleb up and then they kind of condense and then they die. And so just with a simple screen, we found a variety. By the way, I should point out, this isn't even our best hit. This is literally the first one that passed our specs that came off the screen. We threw it on cells, and right out of the gate, this is the sort of effect size that we're seeing. So I again apologize. I can't talk about any of the iterative work we've done since then or any of the other hits that we found in the screen. But I hope you guys can appreciate, again, better biophysical assays lead to better hits. The stuff that we're getting off of our screens that are going after these different pathways are even working in cells right out of the gate and with a profound level of efficacy in a highly targeted manner. And that's something that we and our investors are incredibly excited about. And then this is just a summary. Again, normal cells and compound at 0 and 3 hours, and then the senescent cells with the compound at 0 and 3 hours. Now, the entire value proposition, of course, of Intoxerine is that this is state-of-the-art. This is where we are. Everyone else is doing fragment-based screens. We're doing full-size. But does this really matter? You know, we're an audience that are interested in health extension and longevity. Is this relevant for aging broadly, or is this specific to kind of just a narrow list of pathways? And so what I did is I went online. There's several databases. This is just one of them, Wikipedia. And this is a database of known protein-protein interactions that have been characterized in the literature. And when I typed in just aging to see what popped up, there were 6,338 aging protein-protein interactions that were described in this database. Now, I'm sure many of those interactions are have effective drugs that have been developed for them. I'm sure a lot of them can be screened effectively with fragment-based screens. But where those traditional approaches are not successful and are not sufficient, we believe that our technology can come in, and who knows what different products we might be able to bring to market and what kind of disease states we might be able to target if we were just able to make the proteins that we already know are known to cause those afflictions. So this is a platform technology we're really excited about. We're exploring a lot of partnership opportunities to spin it out, both with mainstream pharma as well as startup companies that are working in the aging space that are of strategic interest to ours. Now, I should take a quick step back and mention that this is just one of Ichor's portfolio companies. Unfortunately, tonight I don't have time to go into any of the other ones or the strategic investments that we've been making, but perhaps Bill and Neil would be kind enough to invite me back sometime in the future to discuss our work on some of the other programs. And with that, I think I'll yield to questions, and thank you very much for your attention.
So we have a few minutes for questions for Kelsey. Thank you. That was great, Kelsey.
Thank you, Neil.
Are there any questions for Kelsey? Over here, Jeff.
Is this available in any form to the average person, in the form of a drug or any kind of treatment or anything like that?
At the moment, no. So the question is this available as a drug that someone could take? At the moment, no. All of the programs that we're working on, though, are things that we're hoping to move into first-in-man studies within 2 to 3 years. So the way I kind of describe it is Bill Faloon is your guy for stuff that you can take right now. Ray Kurzweil and Aubrey de Grey are the guys for things you can take 10 to 20 years from now, and we're really focusing our attention on things that we could move into first-in-man studies on a 2 to 3 or 2 to 5 year sort of time horizon. So these are in active development. They're all well funded, and we're just plugging away through the process.
Good question, Jeff. Here's a question in the back of the room.
Hi, thank you for a great presentation. How far away would you say we are from human trials?
It depends on the program. So some of our programs like Octus Biologics, we just received our first seed funding for about a month or two ago. Other programs are a lot more sophisticated, and we're probably on the order of, I'd say the shortest one is probably 3 years, maybe a little bit sooner, and others are sometime longer than that. It's very program specific.
Okay. Any other questions for Kelsey? Back here. Joel, hold on a second. Let me bring the microphone so we can get this in recording.
I was just curious, is this a separate independent company, or do you work with pharmaceutical companies as a company under their aegis?
A little bit of both. So the general business structure is that Ichor Therapeutics owns our building, owns 13,000 square feet of facility, employs over 30 employees, and has all of our infrastructure. And then Ichor spins out IP portfolio companies. So the way that most drug companies work is they exist as Delaware incorporated IP holding entities, and they work virtually. So they contract with contract research organizations to actually do the work, which is also part of why the process is so expensive and slow. I don't really believe data unless it's generated in my lab. So through Ichor, we've brought all of that in-house. The portfolio companies are managed by my management team. The work is done by my scientists. And then we, at appropriate times, partner with either traditional venture capital or pharmaceutical partners to take the reins and continue developing the programs. So it's a little bit of a program-specific thing, but we do both. We have pharma partners, and then we also have traditional venture capital-backed entities.
Excellent. Where do you see yourself in 5 years, Kelsey?
Neil, I don't know where I see myself in 5 days. And 5 days ago, no, the overlying goal of Ichor Therapeutics is really to be able to accelerate discovery and development of pro-longevity therapies. And we hear a lot in the investment space about the valley, the proverbial valley of death, and how companies die in there and everything else. And really, what we're trying to do is straddle the valley of death and have everything that's necessary, from high throughput drug screening equipment to GMP manufacturing capabilities and GLP toxicology capabilities, to the ability to design and execute clinical trials. And we're looking to be able to do all of that in-house. I'm hoping to have all of that, including GMP pilot manufacturing facility for new things that are identified through our program in mid to late 2019, and our first run of GLP studies so that we can do FDA-compliant tox studies are currently ongoing to show that we have the infrastructure for that. But the idea is anyone that has a program that is in the space that needs the support and infrastructure to push it through the pipeline, the Ichor itself is intentionally project agnostic because, as you guys saw with the classes of damage, we need to have a lot of different products in very diverse areas to overcome this problem. And so we're trying to set ourselves up as a generalist shop that can move things to address all of those.
Have you came across any cures for glaucoma or a way to regenerate the optic nerve?
I haven't. The only things that we're doing in ophthalmology currently are with age-related macular degeneration and with Stargardt's disease. Haven't done a whole lot with neurology or glaucoma. As many of you probably know, age-related macular degeneration is the primary cause of vision loss in people over the age of 50. We think it's caused by the accumulation of vitamin A junk in the back of the eye. So we have an enzyme therapy that we're developing that is able to go into the target cells where the junk accumulates and clear it, and that project is continuing to be developed, and so that's moving forward, but I don't have anything in glaucoma specifically, just that one for eye diseases.
A question from Kathy. Kathy.
Thank you. So I'm a bit confused. You're going generally forward on a variety of different targets.
Yes, ma'am.
Are you priorly, no, wrong word, but are you aware of what causes what, or are you just kind of groping to see?
Well, that's an excellent question. So are you kind of blindly going at everything, or is this slightly more targeted than that? And the answer is both. So in order for a pharmaceutical or a biologic to be viable from a drug discovery perspective, it has to go after a traditional, well-accepted, understood disease. There is no such thing as aging as a disease per FDA. And it's also difficult because if we were to cure aging tomorrow, how many patients would I need to have in a clinical trial, and how long would I have to follow those patients to know definitively that cure it, not talking about extend life 20% or something, cure it. How long would I have to follow that cohort for to know whether or not I've actually done something? So those sorts of studies are the reason why Alzheimer's trials, for example, are so expensive. And so the way that we've been framing how we're approaching aging is very pragmatic. I find diseases that have what we believe very clear targets to go after. They have a very conservative indication or disease that targeting that thing could treat, but then it also has the side effect of mitigating one of those major types of damage. And in that way, we have a viable business case. We can bring in mainstream funding from sophisticated investors, but at the same time, we're checking off those, those damage check boxes to make a play on actual anti-aging interventions. I should also mention we are aggressively exploring the possibility of doing clinical trials in companion animals. We do have a lot of relationships along those lines that could support that. So one could imagine, for example, a pipeline where we do all of our preclinical work, we move into human clinical trials for the conservative disease, but then we can do anti-aging studies in very old companion animals and potentially mix and match different types of interventions to see if we target multiple types of, of damage, what is that going to do? And we don't have firm plans along those lines yet, but that's kind of how we're approaching the space.
A question from John over here.
Yeah, Kelsey. Of all the projects that Ichor is underwriting, which one, or particular one, are you most excited about, and why?
Well, I'm obligated to say I'm actually excited about all of them. And to be very honest, the ones I'm most excited about are the ones that are a little too far-fetched for me to even start yet. But from the projects that we have ongoing and funded, they each kind of have their own advantages and disadvantages. Our macular degeneration program is probably our furthest along and probably the first one to enter the clinic. I'm very excited about that program for two reasons. One, it's an amazing, humanitarian and business opportunity because it's such an unmet need. Second, it's the very first therapy proposed by Aubrey de Grey and SENS Foundation. So if we move that into a viable clinical product, that kind of shuts up all the critics of his that he's been dealing with for the last decade or so. I'm very excited about our small molecule program because we have a very interesting technology that solves a major technical problem that a lot of other people can't overcome. So I think that makes us able to have a huge impact with other people that are, that have their own targets, and there's certainly more pathways than we could potentially drug ourselves. And then the antibody mimetic technology, which I haven't really talked about at all. Basically, we get antibody infusions to target different types of disease, and the disadvantage of that is that they're expensive, you have to inject them, and they're very expensive and unstable. We have a technology that can bind clinical targets in a similar way to how antibodies work, but you can eat it and it can be engineered to go into the bloodstream. And because it's stable in the gastrointestinal tract, there's a lot of age-related diseases that were historically thought to occur in one part of the body, but actually occur in the GI tract. For example, the example I like to use is alpha-synuclein with Parkinson's disease. So alpha-synuclein is a precursor for loss of dopaminergic neurons in the substantia nigra, which drives Parkinson's. It took me like a week to memorize that. And it used to be thought that the alpha-synuclein actually was, created in that area of the brain, but it turns out it's made in the enteric nervous system of the GI tract and migrates by way of the vagus nerve. So actually the GI tract may be where you want to target that. So there's a lot of kind of interesting applications of that technology. So I'm very excited about all of them, but for very different reasons.
One more question over here, Kelsey.
You mentioned about macular degeneration and vitamin A. What about the high-intensity LED lights and the blue light on our screens of our smartphones and iPads and so on?
Excellent question. I do not have an opinion on that or know anything about that, so I'm probably not a good person to ask. Sorry.
All right. Ladies and gentlemen, Kelsey Moody, thank you.
Thank you.
Good job. We've got a lot of tape up here. Our next speaker has been involved in age reversal his entire life and is always on the cutting edge of medical technologies and new advancements. Let's welcome the founder of our church, Mr. Bill Faloon.
Thank you, Neil. You know, Kelsey Moody is an example of what a relatively small amount of money contributed about 8-9 years ago by one of our nonprofit foundations, what that has yielded. You just saw just a small sampling of what he's doing, but his company may find a cure for biological aging. It was a relatively small amount of money, and it was a relatively small amount of money that started the various charities and other organizations that I'm involved in that are seeking cures for the killer diseases of aging and a cure for aging itself. I'm opening up my presentation, interestingly, because I had no idea what Kelsey was going to talk about, with some rather grim statistics for most people in this room, and it deals with our risk for cancer. And when we're young, cancer risk is relatively low, but unfortunately, as we age past 50, the risk, it increases quite a bit. So most people in this room are at these very high percentage risks of developing cancer. Now, if you were a young billionaire and you realized you were going to age into these limits where cancer became prevalent, where every year you grew older, your risk of cancer increased, well, what would you do with your money? I mean, you've got a 41% chance of contracting cancer, and contracting it is one part. The other part is the horrific therapies you've got to undergo. The surgery, the chemo, the radiation, the hormone ablation. There's so many toxic therapies, and they work to an extent, but you don't really feel very much the same after all those horrific therapies. Well, here's another individual who thinks like the people in this congregation do. Sean Parker, very young, multi-billionaire, net worth $2.7 billion, 38 years of age. Well, he was sagacious enough to think, well, what are my problems in this world? I've got $2.7 billion, I'm young, well, I'm aging, and I'm facing an increased risk of cancer with every single year that's added to my life. So he not only put money into a cancer research project, he set up his own institute to investigate a novel way of using CRISPR gene editing technology as a way to potentially abolish cancer as an indignity that the human race has to contend with right now. And unlike wealthy people in the past who might contribute money to the American Cancer Society, he got involved. And as reported in Wired magazine a couple months ago, he is working with a brilliant researcher to figure a way to vanquish cancer, putting his own money, which only represents about 8 or 9% of his current net worth. So the $250 million he put in, it's not going to break him, but it could lead to a major advance in cancer treatment. And that has a heck of a lot to do, by the way, with controlling aging. Because if it wasn't for cancer, I would be advocating that most people in this room undergo at least periodic growth hormone replacement therapy. Our concern is too much growth hormone may increase cancer risk. So we have to let our organs atrophy as a result of a growth hormone deficit because we're just concerned about cancer right now. People like Sean Parker recognizing that, putting a substantial amount of money, but a rather trivial amount compared to what he's worth, into this cancer research. Now, a number of other billionaires worth a lot of money, as far as we know, they did not do this. And if they did, it was to the conventional cancer research organizations who produced very little in the way of meaningful progress. Fred DeLuca, founder of Subway, worth over $3.5 billion. He died a while back at age 67 from leukemia complications. If he had put money into the right type of cancer research, maybe he'd still be around. Maybe the founder of Staples would still be around if they directed their contributions and their investments in a way that would result in a meaningful extension of the human lifespan and the eradication of something like cancer. Now, Kelsey talked about a technology he's working on that will eliminate senescent cells from our aging body. And as most of you know, senescent cells are accumulating in our bodies as we grow older. And these cells impede organ function, they generate chronic inflammatory reactions, and they emit these protein-destroying enzymes. They're called metalloproteinases, and they literally degrade healthy cells throughout your body. So they shorten your lifespan, and they increase your risk of all kinds of degenerative illnesses. So we need to remove senescent cells, and we do that with senolytic compounds. And what Kelsey is talking about is fascinating. He may have an effective senolytic compound right now that could spare us all from the toxicities inflicted by senescent cells. But the bottom line is, we don't have access to Kelsey's technology yet. What we need to access are effective compounds that selectively destroy senescent cells. And the great news is, they exist already in the form of a leukemia drug called dasatinib and a dietary supplement you can buy anywhere called quercetin. And what's really neat is you only need to do 2 doses, perhaps every 6 to 12 months, to remove the senescent cells in your body to a sufficient degree to obtain the results you're looking at on this slide. This was a study published in year 2015 where they gave dasatinib and quercetin to older rodents, and those rodents had a systemic rejuvenating effect. Organs were regenerated. All kinds of problems that were occurring as a result of their age, they disappeared because they were removing these senescent cells with existing compounds. We don't have to wait for Kelsey's therapy to be approved by the FDA. We can start removing senescent cells from our bodies today with effective compounds. And then when his compound gets approved, well, that might be a lot better, and then we can gain access to that also. Now, the fact about senescent cells shortening lifespans, creating degenerative illnesses, that was published by the American Medical Association, September 17th, 2018, just about a month and a half ago. They were talking about if we can perfect the removal of senescent cells using senolytic compounds that Kelsey was talking about and I just talked about, we can transform medical practice. We can enable people to live a lot longer, a lot healthier, because we won't be burdened with these senescent cells that create metabolic havoc. Now, these are a group of mice. These are examples from a group of mice, and one served as a control, which is the typical person in this country. They do virtually nothing about aging, and, well, their spines bend, they lose hair, and they develop cataracts. So the mouse on the left was part of the control group, and they normally aged. But when a senolytic compound was administered in midlife, that group to the right, that mouse, look at how younger it looks. It has a healthier coat of fur, the spine is straight, and there are no cataracts. And other indicators of aging were far improved in the group of mice getting the senolytic compound. This is a remarkable finding. These pictures were obtained from CNBC and was based on a study published in 2017. And that study showed that these animals lived longer if their senescent cells were removed. And so we tried to do some extrapolation from the study as to what that would mean if an 80-year-old person underwent senolytic therapy using dasatinib and quercetin. What would that mean? Well, a person scheduled to die at age 80 might make it to age 100. That's what that lifespan increase might represent. And the great news is we have access to these compounds right now. We don't have to wait for the FDA to approve them. And a lot of people ask me about their failing kidneys, their failing hearts, a lot of medical issues that they're undergoing, and a lot of that may relate to the fact that they've accumulated too many of these dysfunctional senescent cells. They're highly metabolic, by the way. They're very active. They're spewing out those protein-degrading enzymes. They're spewing out signals that ignite inflammation throughout the body. We want to get rid of those cells. And the bottom line is Mayo Clinic, Scripps Institute, a number of prestigious organizations are investigating senolytic peptides, senolytic drugs, ways to purge our bodies of the accumulated senescent cell burden. Now, Jeff Bezos, very interested, founder of Amazon. Jeff Bezos is worth a lot of money. He is putting substantial money, along with Peter Thiel and other billionaires, into senescent cell research to figure out if senolytic compounds can be developed to selectively remove them from our aging body. Now, one of the issues I have with Jeff Bezos and a lot of other of these young billionaires is I respect the hundreds of millions of dollars that they're putting into age reversal research. I have a tremendous amount of respect for it, but they don't have the degree of passion or dedication that we do in this congregation. Jeff Bezos is around 54 years of age. He is not quite as desperate as I am at age 64. In fact, his priorities right now are space travel, space exploration, which is admirable. He puts about $1 billion a year into space exploration projects. Fantastic. I love the technology, but I want to be around to see all that. So he has put a lot of money into Unity and some other projects in order to accelerate the technology of senolytics, compounds that selectively remove senescent cells from our aging bodies. And this is his medical genius. This is the individual who is doing the primary research at Unity, and they're hoping within the next couple of years to have a drug approved that will remove our senescent cells. This is spectacular news. But as we opened up the sermon with Neil talking about one of the people at this church, well, they're no longer alive. And I hate to say it, as the years go by, people in this group will no longer be alive if we do nothing. We need to bypass these conventional people. We respect what they're doing, but they're not advancing the technology fast enough. And to emphasize the significance of the senescent cell burden, Time magazine reported on some research emanating out of Mayo Clinic and the Scripps Institute, where they found that if you just had one senescent cell in a group of 7,000 to 15,000 healthy cells, that one senescent cell would ignite degenerative aging, and it would cause animals to die sooner. But the great news is, if you use senolytic compounds, the ones that we propose to use now, dasatinib and quercetin, the animals lived 36% longer. Wow. 36% more time. And these were middle-aged animals that had their lifespan extended, and some of the animal groups were actually older in the age range of 75 to 90 human years. LA Times reporting on the same research, and they brought out the fact that some of these animals, given the senolytic compounds, they were 75 to 90 human years of age, and their lifespan extended 36%. So we're looking at people potentially who are supposed to die around age 80 living to over 100 if these animal studies translate into human results. We're very excited because we've already helped fund some senolytic studies using dasatinib and quercetin on osteoarthritis patients. And while the experts are telling us to wait, saying that let them do more animal research before we study this in humans, people like myself who are quite desperate to reverse aging, I've helped fund some of the research that enabled us to identify dasatinib and quercetin as being safe and effective in reversing osteoarthritis. This is the preliminary data from a human study. The criteria to qualify is that you needed to be old and you needed to have bone-on-bone osteoarthritis, serious pain, serious joint immobility, and just 2 doses of the dasatinib and quercetin. We spaced them out a week apart. A dose of dasatinib, quercetin week 1, and a similar dose week 2. And the people responded favorably. 90% had almost complete relief of pain and improved joint mobility. And about 6 months later, the pain started to return, which makes sense. Senescent cells form all the time. So the people want us to redose them so that they can get that pain relief benefit again. Now, within a month or two, we're going to have the follow-up MRI data, because we did baseline MRI tests of their joints, and these joints had no cartilage. They were osteoarthritic. They were on bone, suffering pain, and if we can demonstrate cartilage regeneration, I'd like to think that dasatinib quercetin will soon become mainstream. So this is the dosing schedule that we recommend that people undertake. We want to make it very tightly controlled based on your weight. In other words, very high dose quercetin and modest dose of dasatinib. If you're a light person, you don't need as much as a heavy person. So we've published this a couple times before, and the challenge is getting our hands on the dasatinib. It is a leukemia drug, and we're trying to get compounding pharmacies right now to put it together. They're running into some regulatory hurdles, unfortunately, so we're being delayed. Some people are looking into sources from other countries, and I hope soon to be able to identify reliable sources. Some people already feel they have identified them. So if you buy the dasatinib in the United States, it's outrageously priced, but bear in mind you're only going to do this maybe once a year, but it's over $2,000 in the United States. Compounding pharmacies have told us if we can get regulatory approval, we can get it down to $200. Just $200 per year, or maybe $200 times 2 if you do it twice a year, and you can purge enough senescent cells from your body to derive the kind of benefits seen in the human and animal studies. And bear in mind, I don't sell dasatinib. That is a prescription leukemia drug. It is a brand name is Sprycel, a generic name dasatinib. We encourage people to self-experiment and report back to us what kind of results they have gotten. I have personally done this, but I don't have osteoarthritis. In fact, I don't really have any chronic pain at all, so I wasn't able to notice anything, but I have seen some nice improvements in a number of blood markers. Now, the people at Google, as you may know, don't want to grow old. They made billions of dollars early in life. They recognize the fact that they are mortal, but they have lots of money. So they put a lot of that money into a research group called Calico, and that's California Association of Life Extension. It's a very interesting group, and what they're doing is now combining the work that they've done in the laboratory with a big pharma company, injected over $1 billion to develop anti-aging drugs. And the problem is they don't expect those drugs to be available for about 9 years. We don't have that kind of time to wait. We need to accelerate what we're doing for ourselves so that we don't prematurely die while a potential cure for aging sits there in a drug company's laboratory, moving very slowly through the FDA's approval process. We just can't wait. We are all running out of time. I turned 64 very recently, and that means I only have about 20 more years to go on average. I'd like to think everything I'm doing would be buying me more time, but that's the kind of false security, the kind of denial that a heck of a lot of people engage in. They somehow pretend like they're going to live to be 95 in great health and everyone else is going to die. Well, I face reality. I recognize my own mortality, and I am engaged in a number of experimental interventions designed to at least slow down, if not reverse, biological aging. And to try to put this in perspective, we think right now we can reverse, at least partially, many aspects of biological aging. But you look at the chronology of smallpox, where it could have been eradicated before the year 1800, at least in most civilized countries, and yet there was a lot of controversy about using the cowpox vaccine. Many thousands, tens of thousands of people throughout England, where the cowpox vaccine was discovered, died from smallpox epidemics. And throughout the world, 300 million people died just last century of smallpox because there was still controversy. Should we really vaccinate everybody? It's unbelievable. Look at this data here. 1900 to 1979. Hey, we were all alive back in those days. And I think most of us thought, well, smallpox, that was a thing of the past. But in most countries, it wasn't. It was wiping out people in epidemic form, just like aging is doing today. 5,000 Americans die every single day from pathological aging processes. We think we can prevent a lot of those deaths, and yet virtually nobody reaches out and says to their doctor, can I do more? Can I do more than just take your prescription drugs that deal with an ailment here and there, and they don't even always work that well. But can I do more? And people don't even bother to investigate. It's very regrettable. We look at penicillin, when that could have been made available almost a century before it came into widespread use. When you think of all the children who died of bacterial infections, and they could have been cured had someone just accelerated the technology. Purpose of this church, purpose of everything I do is to accelerate age reversal research so we can all live long enough. Take advantage of Kelsey Moody's technology. Take advantage of all this research that's going on right now. Citrus, it cures scurvy, and yet it took 400 or 500 years for it to be accepted. Lots and lots of people died needlessly, just like they're dying needlessly today. And this statistic was sent to me by email by one of our supporters, and they brought out the fact that 4 or 5 years after citrus was proven to be a treatment for scurvy, to prevent and or cure it, over 130,000 British soldiers died of scurvy. None of them had to die, but they weren't willing to take a technology that had been demonstrated in a clinical trial to work and use it. And as a result, 130,000 people dead. It's ridiculous. Now, last night, I had the privilege of talking to a medical doctor who has developed a way to potentially treat early stage Alzheimer's, reverse it, get people back into a normal living pattern, restore functional independence. And you heard about this technology in this church in year 2015. A group of charitable women, their group was called Sharp Again Naturally. It's a nonprofit group. They go around and they talk about the fact that the media and doctors say you can't do anything about Alzheimer's. And guess what? If you treat an Alzheimer's patient individually, you look at what they need to restore cognitive function, you're getting results. Just last week, published paper came out showing the results of 100 Alzheimer's patients treated with the technology you heard about in this church. In fact, this group of medical people, they're working with the Sharp Again Naturally charity to enhance the ability to reverse cognitive dysfunction that leads to Alzheimer's disease. And 100 case studies are contained in this report, and we've reprinted that, and we have that outside our doors for you to pick up tonight in case you are developing mild cognitive impairment or know somebody with Alzheimer's. You don't need to believe your doctor or the media. The good news is if they look at themselves as an individual and correct the toxins, the deficiencies, the dietary inappropriateness that they may be engaged in, they may be able to regain normal functionality. They may be able to drive their car again, live independently, button up their coat. They may not need full-time nursing care. This study, again, published 1 week ago. You heard about it in this church 3 to 4 years ago, and it's been known in the scientific community to an extent that if you really want to do something to reverse cognitive impairment, you can do it. But you got to make some effort. You got to dig into the scientific literature and find out what is causing you to lose your mind. So I'm going to conclude with a slide you've seen before. Benjamin Franklin, he predicted at some point that all the diseases that mankind encounters, including that of aging, will at some point be cured. We won't have to suffer aging any longer. And his prediction is coming truer almost every single day. We find a new advance that enables us to better prevent or treat a degenerative illness. What you heard Kelsey Moody talking about indicates that within the next 5 years, his company may be bringing out breakthrough therapies that can enable us to live a real long time. But in the meantime, we've got to take advantage of what we know now, and that is implement what we can do as it relates to reducing our calorie intake, improving our ability to be physically active, and to not let the typical Western diet, the typical Western lifestyle be the factor that causes us to prematurely die. So I thank you all for attending, and I'll be happy to answer any questions.
Thank you, Bill. All right, any questions for Bill tonight? Let me go over here to this one. I have a question from John. Bill?
Hey, Bill. I came across a study, albeit limited, that seemed to indicate that metformin may slightly suppress testosterone. It's not going to stop me from taking it by a long shot. Would you recommend exogenous testosterone when taking metformin?
Yes, I do. There's a couple studies that indicate that men who take metformin, which is an anti-aging drug we recommend everyone take, by the way. Metformin, we've talked about it extensively in this church, but it may reduce testosterone production. So the easy way to get around that, since as you're growing older, you usually need to replace your testosterone anyway, is, well, if you have a blood test showing your testosterone is low and you're taking metformin, simply have a doctor prescribe compounded testosterone cream. You can get it from a compounded pharmacy at a reasonable price, and you just rub on the cream every couple days to restore your testosterone levels back to normal. Now, women with polycystic ovary syndrome, they produce outrageous amounts of testosterone. It causes them to become overweight. It creates all kind of metabolic problems. Metformin works to lower that testosterone in women, and is the single most effective therapy we know to treat polycystic ovary syndrome. And that's been known in the scientific literature for decades, and yet most of the practicing doctors that we run into are prescribing the wrong medication. They're prescribing high-dose estrogens to women who already have too many hormones in the form of testosterone. So, John, you're correct. For some men, testosterone will be reduced in response to metformin, and the easy way around that is simply use more testosterone.
There's a question over here.
The same thing about metformin. Right now, so many doctors, even Dr. Oz, is against metformin. For diabetes, they say the side effects are terrible. What happens then? Are they all wrong?
Yeah, they are. I've been on testosterone. Well, they're just wrong. They're just wrong. You can go to PubMed.gov. That's a website that our federal government uses the tax dollars in a good way for, by the way, the National Library of Medicine. It pretty much contains every published study around the world. Type in metformin and aging and stay up for the next 2 nights reading all of the studies showing that metformin may slow and reverse aging. Type in metformin and cancer and see all the studies showing that metformin may prevent and or treat many forms of cancer. Show how metformin can prevent the progression of prediabetes into full-blown diabetes. Dr. Oz, unfortunately, came under a lot of criticism in 2010, 2012. He used to recommend a lot of the organizations that I'm involved in, and unfortunately some other organizations that he probably shouldn't have. And he got into some trouble. So he's turned, I believe, rather conservative. So he's telling people take fish oil, a multivitamin, and I think one other thing, and that's about it. So he is not someone who's going to lead people into a significantly longer lifespan if he's going to stay that conservative, because there's only so much a multivitamin and fish oil is going to do. I think CoQ10 might be one of the things. There's 3 things that he recommends people to take, and that's about it nowadays, and that's unfortunate that he came under some criticism, and some of it was just overblown.
There's a question over here.
Must the 2, dosanitab and quercetin, be taken together, or can one be taken separately without the other? And when you said it improves kidney function, in what sense does it improve it? Does it decrease creatinine and BUN levels, or does it just In what sense?
Okay, you have to take the dasatinib and quercetin together. They have synergistic properties. If you took either one alone, you wouldn't get the benefit. So you take the high dose at the identical same time, and what we have with studies, with just animal model studies, are indicative of improved renal function. We don't have the human data yet. So if you're suffering severe end-stage renal failure, I'm not sure this is going to reverse it, but it is something interesting to attempt, because when people approach me with chronic kidney disease, I don't have a lot of solutions for them if they're already following a healthy lifestyle. Now, if they're following the typical Western unhealthy lifestyle where they're eating lots of red meat and they're not doing anything as it relates to coenzyme Q10, I can give them some suggestions to improve their diet. Suggest they try to transform into more of a vegetarian, plant-based diet, and that can sometimes improve renal function. But the data on senolytics and kidney failure, not as strong as we'd like. We simply need to do more studies on it.
Question over here.
Hi, Bill. At RadFest, you were talking about NAD infusions and the introduction of patches. You said there might be some research that came out on it.
Yes, and good news on it. Yes, absolutely. I think you've heard me talk in this church many times about the virtue of restoring NAD plus levels back to youthful ranges. And over the last couple years, the only way we knew how to definitively do that was through infusions, where you go to a doctor's office, spend 4 to 6 hours with the NAD dripping into your veins, and it was a rather unpleasant process and a rather expensive one. So we funded a small clinical trial where we used patches to see if we could get the NAD to absorb through the skin. And what we found is that the patches were boosting NAD plasma levels the same way that NAD infusions were boosting NAD plasma levels. So this is spectacular news for a lot of reasons. The infusions were costing some people $4,000, $6,000, $7,000. The patches can be obtained from Archway, a compounding pharmacy, and probably a bunch of other compounding pharmacies. Archway is in Louisiana. I believe they're selling them now, 6 patches for $99. Just $99 for 6 patches. Bear in mind, I don't sell this stuff. I'm just telling you where you might be able to get it. Archway, $99. And those 6 patches might be equivalent to several infusions. That may be all you need to restore your NAD levels. Now, you need a prescription. So you need to find a doctor who will prescribe the patches, and we are amalgamating doctors locally who are going to be knowledgeable about these patches so they can prescribe them properly and teach you how to put them on. So the great news is, this news is only about 2 weeks old, that the patches appear to boost NAD plasma levels as well as the infusion. So we're going to save a lot of money and be able to get a lot more people to restore their NAD plus levels.
That's great news, Bill. Here's a question from Andrew.
I've been reading about trace minerals lately personally, and was wondering why a lot of people don't talk about it, because our bodies are basically water and trace minerals. And apparently a lot of most Americans have too much calcium, and the body doesn't know what to do with the excess calcium. Is there a chelator? Is there some way to get rid of it? And is it causing an epidemic in this country?
There's mineral imbalances that people have where they have insufficient magnesium and a number of trace minerals, and they sometimes overindulge in calcium as it relates to their dietary choices. And they don't do anything to target the calcium in their body away from their arteries and into their bone. We don't talk, or at least I don't talk a lot about nutrition at this church, because people read a lot of the publications that I have and they already understand that. And we have other speakers who come in here and talk about that. I try to talk about the age reversal research that we're actively involved in. And the only place you're going to really hear that is either through this church or through the rescueelders.org website. So we are really a privileged group of people. We interact, we exchange information, and we find out data often months, if not years, before the general public.
A question back here.
I found out recently my mother has heart failure. She also had pneumonia many years ago, which has affected her breathing ever since. I put her on CoQ10 as well as the pharmaceuticals that she's already on. She's on 4. She has high blood pressure too. So Liposil, Metoprolol, and amlopine, as well as the furosemide. So I have her on CoQ10. I was at your store tonight, and I also to supplement it with super selenium complex and L-carnitine, as well as the super ubiquinol CoQ10, because I had the Frank Sinatra CoQ10 from years ago that I had started on. So I was wondering, and I'm going to call in the specialist, wellness specialist tomorrow morning to talk to. I was wondering if you have any additional comments or recommendations.
Well, everything you talked about is good. It's information that's been published now for the last 20 plus years. So everything you're doing is good, but with the innovations that are occurring in the field of regenerative medicine, I'd like to think, and the American Heart Association even thinks, that if you restore her NAD+ levels, that may be beneficial if she's suffering congestive heart failure. Might be beneficial. Also, removing senescent cells, I would like to think, might be beneficial. So there's a number of treatment modalities now that go beyond everything you just described. And I showed, I think last service, the American Heart Association's interest in investigating the potential role of elevating cellular NAD+, restoring the energy output of heart muscle cells so that perhaps congestive heart failure patients can recover a lot better. So you're doing a lot of good things, but I think some of these newer treatments might be more effective.
I actually brought all that list up to her doctor, and he said to wait on that and to get her heart stronger first with the lisinopril that he put her on with, as well as furosemide, because her legs were all swollen. He said to do that first and then to look into that afterward. So probably, thinking what I should do is then find a way to still get her in.
Well, your doctor's correct, because see, the fluids that we that we previously needed to put in her to boost NAD would have put a stress on her heart and her lungs. But the good news is, with the NAD patches, we don't need to use the infusion therapy to increase substantially her NAD+ levels. So this advance that was funded with a relatively small amount of money, it yielded a really important piece of information for people just like your mother, who can't tolerate high volumes of fluids dropping into her body because she can't clear them efficiently. With the patches, you don't need the fluids.
Question from Kathy.
Thank you. Hi, this was the first time I'd heard of the Disinitab, and while you were speaking, I did a little Professor Googling on it. It has serious side effects. Are you talking about a particular quantity of it?
Exactly. Yes, dasatinib, a leukemia drug that leukemia patients unfortunately take every single day. Typical leukemia patient takes 36,000 milligrams a year. Okay, 36,000 milligrams a year, and it does cause side effects along with their leukemia, creating complications. We're advocating about 400 milligrams a year of dasatinib for the typical person seeking to use it as a senolytic. There's a big difference between 400 milligrams and 36,000 milligrams. So you're correct. It's a dose-response relationship that makes it very safe to use dasatinib based on everything we know so far. The dose is trivial compared to what the leukemia patients have to bear.
Ladies and gentlemen, Mr. Bill Faloon, thank you. All right. Great presentations tonight by Kelsey and by Bill. Thank you for your presentations. Really good job. I'd like to remind you that next month we'll be here, December the 13th, Thursday evening at the same time. Please bring a friend. And now we have a dinner scheduled for you downstairs. If you'd like to join us in a reception for Bill and Kelsey, we're going to have some delicious food, so stick around. It's great to see you all, and we'll see you again in December. Thank you for coming.